Professor Thomas Jentsch is researching a process that is vital to all organisms: ion transport across cell membranes. This is how, for example, nerve cells transport signals, or how water and salt are transported in the body. The ions can be chloride atoms with a negative charge or potassium atoms with a positive charge. Special proteins in the cell membrane, so-called ion transporters and channels, control the transport of the ions. If the channels are obstructed, this can result in serious diseases. We are interested in ion transport processes across cell membranes, and we systematically take specific gene families as a basis. One of these gene families encodes for chloride channels, and we have identified this family for the first time. Other families we are studying include potassium channels and co-transporters. We examine these processes on various levels, from the molecular level to proteins, structure-function relationships all the way to cellular biology, and, above all, diseases and pathologies that are caused by these processes failing. This is how Jentsch and his team, in cooperation with other research groups, have managed to explain human genetic diseases that are caused by a malfunction of ion channels. These include, among others, osteopetrosis, an illness in which an increased ossification can be observed in comparison to a normally developed bone, as well as a kidney stone disease, a genetic type of deafness, a hereditary type of epilepsy, and myotonia, a hereditary form of muscle stiffness. Among others, Thomas Jentsch and his team use frog oocytes to examine how ion channels function and how mutations influence their electrical characteristics. These cells are particularly big, and it is therefore easy to manipulate them. In addition, after injecting the encoding RNA, they produce a large number of channels whose characteristics and interactions can then be examined.
We are injecting the RNA that encodes for an ion channel together with the RNA of another protein. And the frog oocytes will express both proteins, and subsequent analysis of the electrical currents will tell us whether both proteins interact.
Jentsch used this method to identify the gene of the electric ray that includes the blueprint for a chloride channel several years ago. Depending on the voltage, this channel allows transport of chloride ions with a negative charge across cell membranes. Such channels exist in basically all organisms. If they are lost, it can cause various diseases in humans. By changing the blueprint and examining the electrical activities, the scientists can therefore find out how the channel functions and how it is regulated. Using the patch clamp technique, they measure highly sensitive currents of individual cells in the tissue.
Wir wollen prinzipiell Physiologie verstehen.
In principle, we want to understand physiology, meaning how nerve cells communicate with each other in a cell structure. We mainly focus on the ion concentration of specific ions in the nerve cell, which is important for the function and the response of these nerve cells.
Wichtig ist für die Funktion und die Antwort dieser Nervenzellen.
What is so special about Jentsch and his team's work is that they combine many different methods: structural and cell biological methods, biophysical and human genetic methods, as well as transgenetic mouse models. Their research goes from molecules to cells and all the way to organisms, and their results are of great medical significance. Ion transport is extremely important for a great number of cellular processes as well as for the organism as a whole. And since ion transport processes exist in every cell and in every organism, our research always takes us to different organ systems, such as kidneys, bone, inner ear, brain, etc., so that we always make new exciting discoveries and are surprised again and again.